Hello, my name is David Keire. I am going to be presenting on the subject of two ICH guidelines, Q2R2 and Q14. Q2R2 is a revision of Q2R1, which is a guideline on analytical procedure validation, and Q14 is a guideline on analytical procedure development. Before I begin, I want to just say that everyone deserves confidence in their next dose of medicine, and that pharmaceutical quality assures the availability, safety, and efficacy of every dose. Pharmaceutical quality is the focus of the office that I work in at the FDA. And what do we want? We want fewer drug failures. We want to minimize and eliminate drug recalls. We want regulatory flexibility and fewer filings. We want to lower barriers for new analytical technology and ultimately quality drugs for consumers. Now, I don't want to get into a discussion about what is the central science, whether it's physics or chemistry or biology. I'm sure many of us have opinions in that. But if you think about steps in a drug's life cycle, from drug development to manufacturing process monitoring and control, to quality control and batch release, and ultimately post-approval surveillance, all of these steps have key decisions that are made based on measurements. Analytical procedure measurements are providing the data that drive decision-making across all this landscape. So it behooves us to have the most reliable and robust measurements that we can get, and that's really the focus of Q2, R2, and Q14. So how do these two guidelines help? They standardize. They provide guidance on the content of sections of the common technical document. They harmonize, depending on how you spell it, and so that there's common requirements globally. And ultimately, together, they provide a framework that's adaptable to technological change and allows for continuous improvement for analytical procedures. So Q2R1 was finalized in the 90s, and a lot of scientific and technological progress has been made since the document was written. There's a lot of advanced therapies that are in drug development and commercialization, and the associated analytical techniques used to make measurements on those therapies are multiplying. There's a lot more use of hyphenated techniques, things like liquid chromatography, mass spectrometry, or spectroscopic approaches that are multivariate. For example, near-infrared or Raman spectroscopy tools. So for Q2, the existing version of Q2, what are the issues and the cost? As I mentioned in the previous slide, Q2R1 is not directly applicable to multivariate spectroscopy data. So with the lack of clear guidance, that leads to inadequate validation data and regulatory submissions, and ultimately to recursive information requests and responses that take time. And things like near-infrared technology is commonly being used for innovative techniques, for example, things like real-time release testing. And so with the current version of Q2R1, there's kind of a barrier to innovation in analytical approaches for pharmaceutical quality assessment, which is one of the reasons why it was decided to work on a revision. So for Q14, there has been no ICH guideline on analytical procedure development. And because of that, applicants rarely present performance evaluations, which they might do as part of an analytical procedure development in regulatory submissions. And that can lead to recursive regulatory communication around non-conventional analytical procedures. For example, things like process analytical technology-driven multivariate models used for process control. And the lack of a guideline and a place for these kinds of data in application impedes applicants from presenting a scientific basis. For example, quality by design data that might allow regulatory flexibility in post-approval analytical procedure changes. And ultimately, this delays access to medication and increases costs. So what is in these guidelines? What is developed is validated, right? So above this dotted blue line that's in the slide here lies Q14, where you use analytical procedure knowledge, can combine with what objectives you have to decide what performance characteristics you want, and you have analytical procedure related information from development. And based on all that information and knowledge, you move into Q2, where you write a validation protocol, you perform experiments, and perform evaluation of data, which you would write up in a validation report. And that provides a snapshot of the analytical procedure performance, and it's kind of a decision point that this procedure is going to work to measure the attribute, the drug attribute that you want to measure. And after that, it moves into analytical procedure lifecycle management, which is, again, crosses back into Q14. So in the previous slide, the Q14 looked like kind of a small component compared to Q2. But really, when you look across the analytical procedure lifecycle, the tan box here is the Q2 part, and all the rest of the things are Q14 elements. So, if you look on the far left of the slide, where you have a drug product where you need to make measurements on, and you have some product and process understanding, and you know that you have some attributes that require testing, you develop an analytical target and you select technology and perform a risk assessment, identify parameter set points and or ranges, define an analytical procedure control strategy, and then you do the validation. After the validation, you go on to routine use and ongoing monitoring. You may have to do some changes to the procedure over time, because the validation is just a snapshot of the performance, and that can change for a number of reasons. I mean, your product may change, other things may change. You may want to, adopt a new technology. And so this slide basically describes what's in this box is all of Q2 and Q14. So I want to take a moment to talk about in a little more detail a couple of the things that I touched on in the previous slide. One of those things is analytical target profile. Now, the analytical target profile is, you have an intended purpose of a measurement, and you have some sort of performance target, and that can be described in this ATP. And that's really a technologically independent profile. You could have that performance, you can meet that performance target and intended purpose by more than one technology, more than one analytical procedure. And so based on your knowledge and a risk assessment that you perform that says, here's the most likely technology that's going to give me the performance I want, there's some risk associated with that. And then you make a technology decision, which leads to an analytical procedure attribute that you can measure, and there's associated criteria with that, and of course all the analytical procedure parameters that go into that need to be considered and are a part of the equation of analytical procedure development as described in Q14. Now, another element I want to touch on briefly is when you need to make a change to an analytical procedure, you have to do a risk assessment there as well. And the risk of that change would depend on the extent of the change to the procedure, which critical quality attributes you're measuring, how important it is, how complex your product is your matrix going to interfere, and hopefully, you can control those risks by, what you know about the product, what you know about how the product was made, the process knowledge, and also about what you know about the analytical procedure, which you might have developed or might have accumulated during development of that procedure. And if the control of those risks outweigh the risks, you can decide to make that change. In addition, you could provide all these kinds of data and analysis of the data, assessment of those data, to a regulator to reach agreement about a reporting category using Q12 tools, which give you some post-approval regulatory flexibility. Now, I won't be able to go into all the other wonderful things that are in Q14 and Q2R2, but I've touched on a few of them, and hopefully I've stimulated enough interest that you will feel the need to go and read the entire documents. And they're available to you because we reached step 4 in November of 2023 after 5 years of work by the expert working group. And we're currently working on training materials as a IWG right now that will hopefully become available in about a year's time. I think in summary, I want to say that together ICH Q14 and Q2R2 describe the development and validation of analytical procedures used for the assessment of drug substance and drug product quality. Q14 describes the scientific principles for development, change management, and submission of analytical procedures using minimal or enhanced approaches. Now, I didn't really provide much touch on minimal versus enhanced approaches, but again, it's available to you in the document, and there's training materials to come. And ICH Q2R2 provides guidance for establishing and submitting evidence that an analytical procedure is fit for assuring drug quality, just like R1, but it also allows, it has more flexibility for innovative technologies or new or different technologies besides HPLC, and hopefully it's an adaptable framework that can be applied to any analytical approach that you might take. I also want to say that Q2 and Q14 exist within a continuum of many different ICHQ documents, quality documents, and we pulled from many of them in the process of putting together the manuscripts. And I just point out a few of them here. Q8 R2 on pharmaceutical development, Q9 and quality risk management, and Q10 pharmaceutical quality systems, Q11 development and manufacture of drug substances, Q12 on pharmaceutical life cycle, and Q13 on continuous manufacturing. All of these things were considered and when we developed our guideline, and they kind of all work together for a common goal of assuring drug quality. Now, this picture was taken in Prague in November of 2023, and you can see how happy everybody is because we've actually just, at this point, we had just signed off on those two documents after a very long period of time working together. But I can't say enough about this group, a really super collaborative, collegial group that I've enjoyed spending time with over the years. And I thank you for your attention.